Can you all hear me okay?
Yep.
Yep, okay, jolly good. I don't know if any of you remember the first time I did this. It was back in 2012, 2014. Anybody remember? Paul Spearman must do, because it was a British bulldog.
I remember.
Sorry?
I remember.
Yeah, you do. Okay. This is basically, I started doing this because I just enjoyed the whole process of brewing. And back in the day, you couldn't get many wet yeasts, anything different. You had very small selection, you know, not even a handful of different yeast you could use. So actually using something like James was talking about, using 1845, which is something I did until I actually used a bottle of Spitfire and made a porter with the yeast, and it came out somewhat funky and sort of odd tasting. So it works some of the time, and I think 1845 is a good yeast for that. And as you say, it clears really, really well because it's a highly flocculent yeast. But the idea was to take one cell from a bottle of beer and then ferment a beer with it. And the idea is that you know, or you hope, that the yeast is actually sterile. There is nothing but yeast in there. So you actually get a clean fermentation from a single cell. One of the flaws with this is that British Bulldog, and Paul can correct me here, is a dual strain, or the yeast they use.
Why is it a flaw, Phil?
Sorry?
Why is it a flaw?
It's a 4 strain.
No, sorry, you said it's a flaw.
No, it's a dual strain, isn't it?
It is a dual strain, but why is that a flaw?
Because if you've got 2 different strains, you're selecting one of them.
Okay, yeah, I got you. Yeah.
So if you've got 2 different characters, you know, one's more flocculent than the other, or one's fruitier than the other, you are selecting that particular characteristic. Yeah. And you might want to do that. You know, that might be one thing you want to do. What the professionals do is, if you've got a brewery who has a yeast that's mutated and they're getting inconsistent results, what they might do is select certain strains or yeast from this, then ferment small batches until they find the yeast that actually gives them the character they want, and then ferment with that. And that's what Hook Norton did. They actually had their yeast examined, they selected one strain from it, and then they started to ferment with that. And that sort of goes back to the whole original idea that people didn't really, well, it's not until 1850 that people really knew what yeast was, so they didn't really know what fermented the beer. It wasn't until the invention of the microscope that people realized what was fermenting their beer. And that's when they started to play around with strains and able to have consistent beer quality, which was important when you're doing mass-produced beer, which, you know, during the Industrial Revolution, that's what happened. The small breweries, the town breweries disappeared, or the individual pubs that brewed their own beer with their own yeast that they had. And they went over to larger manufacturers that had money behind them and consistency in their beer. Okay, let's start this. So, yes, as I said, back in the days, you couldn't get more than half a dozen strains. And it wasn't till the late 90s or the 2000s, something like that, that you were starting to get Y yeast coming through. Otherwise, you know, you would have to go to a bottle of beer. And I've heard of lots of people making good beer from buying a bottle of beer and using that, as James has. So let's see. The first book that got me into this was this book here, First Steps in Yeast Culturing by Pierre Rogeau. Part 1, it says there. In fact, there was never a part 2. So they never actually produced the second book. I have got a PDF of this. Dave House actually copied it. I had a copy, he borrowed it, put it on a PDF at work, and we can post it somewhere on the lab, whatever. I'll give it to Daz and then he can post it on there. It's got quite a few interesting pictures. Other books that have about yeast culturing is Fix and Fix, Analysis of Brewing. And obviously there's a whole chapter in the yeast book, about 70 pages on working with yeast and preserving yeast and doing all sorts of things with yeast. I think I've read some of it, but I haven't read an awful lot of it. So why would you want to save your own yeast? So the whole idea is to grow yeast cells on an agar plate, which multiply from a single cell and taking one group of these cells for fermentation. Or you could, in case of choosing the wrong cell, because we're not going to make lots and lots of starters of lots and lots of separate yeast, take several of these clumps and use that. So they could all be the same, or they might not be. We don't know, because there's no way of finding out for the home brewer. So you're going from one cell to billions. Good evening, Mark. Reasons for doing this, as we've said, you couldn't get many yeast, potential infection in the bottles. You've done it now a couple of times, have you, James? Have you found that you've got infections at all?
I grew up through multiple steps, but I've only brewed with it once.
Okay.
But one of the last things I did was I did a large starter, and then crashed it out in the fridge. So I had very basically solid yeast slurry and then clear sort of beer on top of it. So I could check that there was nothing sour or nasty in it before I pitched it. But that's all I did really. I mean, I was just winging it compared to growing up the cells properly.
Well, and if it works, why not do it? You know, Fuller seem to be pretty reliable with their beer. Another thing is you can keep your own house yeast. I'll talk about that if you want to do that on, you know, to keep it on slopes or slants, whatever you want to call them. And it's a bit of fun, you know, it's something that you don't normally do. But these days, let's face it, we've got hundreds of different yeast. You know, looking back, we had a choice of about half a dozen, if that, and they were all dried yeast. And now, you know, I looked on Malt Miller today, and there are over 100 different yeast, and I'm sure there must be a couple of hundred, something like that. You know, compared to even sort of 5 or 10 years ago, you know, there weren't that many. There were white labs and Y yeast and the dried yeasts, but now, you know, there's so many about. So what's the basic equipment you need? You need a pressure cooker, autoclave, Bunsen burner, loops of various types, glass containers, malt extract or wort. What I used to do was collect some beer and sterilize that in the pressure cooker. And so, you know, I didn't have to make up my own wort. And you obviously need some agar to set the slopes if you're going to use those. And Petri dishes or plates, you know, plastic or glass, I had both. Okay, why do you sterilize? Because bacteria survives, well, the bacteria doesn't survive the boiling, but the spores do. So it's all right if you're boiling your beer and then you're adding the yeast, the pH changes, the bacteria can't grow if there are any spores from the bacteria in there. So you just need to keep it at 121 for 10 to 15 minutes. And that's the best way to do it. And this is what you need, just one of these ordinary pressure cookers. Autoclave is a bit more complicated and more expensive, but the pressure cooker for 20 minutes. And what you would do is, I seem to have got in the wrong order. Let's see if I can, what am I looking for? No, okay. What you would do is take your dregs from a bottle, let them settle out, put that into some boiled water or sterile water, and get that into suspension. And then you would take a loop and take some of that, or just a very small amount of that, and spread it on a plate, which is one of these things here. And that's what you would do, is to spread that on a plate, and it has to be diluted. So what you need, sorry, going back again, I should have practiced this a little more earlier, but this is what I used to use to have a safe, clean area while you're working. You can open up the plates, you can open up the vials, and you can transfer from one thing to another in the small area around. Bacteria is on dust particles, so you really want to do it first thing in the morning, something like that. You don't want your windows open and the gale blowing through, otherwise you've got dust everywhere. And you've got different sorts of loops. They're the two loops that I've got. This is the equipment. You've got plastic plates, which you buy already sterile. You've got those little screw top bottles. And you've got the large containers at the back. To sterilize your wort, your water, or to make slopes, you use your pressure cooker. And you can see on the right hand window there, you've got little bottles of sterile water. This was just boiled water. And you've got different size containers with beer in. You can make slopes the same way. You just, when you pressure cooker them, you put them in at an angle using foil, and as it cools down, it sets. So then you've just got the lids on just lightly, and then once you take the lid off, once it's cooled down, take the lid off and screw the tops up. You know, nothing will get in there at that point. I've still got some agar somewhere. That is probably a good 10 years old. And to make a plate, you put the agar, it melts at 95 degrees, as it said, and it solidifies at 45. As you can see, the deliberate mistake on the right, you can see the agar is starting to solidify as I pour it onto the plate. Which does make it difficult if you're trying to street wipe the loop across the plate. It's gonna catch on any bumps. You want to spread the yeast finely, and I'll show you why. So we've really covered that. So here we go. You've got the yeast here that you fermented in a small amount of wort, and then you shake it up a little bit and you put it into the sterile water. So you've just got a couple of drops in there, and then you sterile transfer onto your loop, and then you just streak it across in that way, in the manner on the right there, and then the yeast starts to grow. And as you can see on the left where it started, I obviously didn't do a very good job because it's only really on the top of the plate, but you can see the individual yeast colonies. They all come from, except for ones that are slightly distorted, they all come from one cell. So the larger cells that are round in shape, not deformed in any way, should be the ones that you pick. Oh, there you go. So these are two. One is undiluted and the other one is diluted. And you can see you can't really see any individual cells, and what could be hiding in those clumps that are all lumped together could be bacteria. You don't know. So you pick out the nice regular shaped cell clumps and one or two or three of them, and then sterile transfer them onto a starter. So you've got your plate with the yeast on, flame your loop, and then you cool the loop because it's going to be hot in the agar, and then you take a colony off. So I've got the lid in my left hand and taken the lid off and flamed the top of the bottle. The yeast is in the safe working area at the moment, or roughly. And then you place it while still in the safe area into the sterile wort, flame the top again, and then replace the lid, which I had in my hand facing down all the time. So it's like a constant flow. It's just a case of getting used to it. And you can go from a small bottle there, takes 4 days. So I've got a 3 litre or 2.5 litre or so starter. And Hook Norton used to send me their starter slant when I went for a trip around the brewery, and I discussed with the microbiologist, and for about a year she sent me the slope that they started their new batch of yeast with, and I could guarantee how long it would take. If I took a few cells off, I could put it in the small one, day and a half later I put it into the next one, day and a half later into the next one, and so on. And it used to take 4 days to get up to that. It was almost like clockwork. And a slope like that you would keep in the fridge for 3 months, and then you're supposed to grow it up again. So you take some yeast off that, grow it up, and then spread it out onto a new slope and keep that in the fridge. So within 12 hours, you've got fully fermenting beer. So from one cell, if you like, from one cell to billions in, I don't know, 24 hours, something like that. So that's it. Seem to have missed a few things out there. I don't know if there's any questions people want to ask.
Can you tell me, Phil, what the little glass vials that, or I presume they're glass, the vials that you had that you were sort of pressure cooking and then using for work beyond that? Do you know specifically what they are? Because I looked around for these a while ago and just couldn't find anything that was going to
Right.
be pressure
I know
cookable.
they use a lot of plastic ones now these days,
Yeah.
sample bottles. But I used to get them from a place called A1. I forget what it was called now. It was actually where I used to live in New Elton. So it was just round the corner from me. But you should be able to get them on the internet.
I've looked and really struggled. The best you can find now,
yeah,
the best I can find now is pre-sterilized, a similar sort of thing, but they're all plastic, so they won't go in if they melt in a pressure cooker.
Yes,
or other ones which are glass, but the lids wouldn't go in a pressure cooker basically. So yeah, when I was gonna,
it's been a long time since I've done it. It's
okay.
I don't know, 10 years since I've done it really. So maybe a bit less. But BrewLab used to use those, so not sure. Could give them an email or send them an email, see if they can.
Lee might know something.
Yeah, well, I would imagine so.
Sterilised plastic ones, you won't need to stick them in an autoclave unless you want to keep reusing.
It was the fact that if they're pre-sterilized, then you then still had to sterilize the wort and pour it into there. Whereas if you could otherwise you could put them in there, put the wort in there with the cap loosely on, and put them in the pressure cooker, and then all you have to then do is tighten up the cap at the end, and you know it's all good. But
Mm.
Last
this was,
micro team up.
And it's more difficult to make your own plates.
well, this was specifically to I was not making plates but slants.
Yeah.
So a similar sort of thing, but
And once you made a batch of slants, they last forever. They just start to dry out a little bit, but they will last for years. So, because they are literally sterile.
Has anyone bought a slant from, who are the places that sell slants?
Brew Lab.
Brew
Do they
Lab,
come glass or plastic?
I've not
I
bought
think
one.
now
from
they're
them.
plastic.
Yeah, I think they were plastic. I got
You-
some at the BrewCon a couple of years ago, and yeah, they were plastic.
Mm.
You used to be able to Brew Lab, phone them up and say, I'm looking for a yeast that's sort of West London beerish, and they would send you essentially the whatever brewery it was. You couldn't
Yeah.
say I want Fuller's yeast, but they had it usually, and you just described in the rough area, and they'd be like, oh yeah, this is the sort of right thing for that beer.
Don't they do that anymore then?
No, they sell through, I think it's Hop and Grape online, but they've
Okay.
only got like 4 or 5 strains, so it's a bit of a shame really, because you used to be able to get a hold of all kinds of yeast from there. But
I suppose that's very time consuming for not much money. That's the
I would have
problem.
thought so. Yeah, I think they're more in the commercial banking and growing up when needed kind of thing. But
Yeah, doing courses, etc. That's what
Yeah, right. I did write to them for the Sussex 1 Harvey's yeast, and they grew one up. It was like 12 quid or something like that.
Yeah.
Send it over.
Well,
Got to say,
one
it
of their
wasn't as nice
strains
as
is
the,
Sussex, isn't it?
yeah, it wasn't as nice as the 1469 though, as everyone finds out.
Still, we as homebrewers are all very precise these days about the number of cells we require to pitch. How do you gauge them? How do you think you know the number of cells that you are growing on each time? Or is it just guesswork?
Guesswork.
Okay.
And it looks healthy. It's fermenting quickly. I was talking to Chris about yeast these days that we get, even the wet yeast, they're so much healthier than what we used to get, I don't know, 10 years ago. You know, a smack pack, you smack it and you weren't sure if it was going to take 3 or 4 days to come up or it'll be up in 24 hours. But nowadays you smack some of the smack packs and they're up in a few hours, ready to pitch.
I had one that hadn't been smacked at all, and it was up. But they said it was all right. Oh no, actually, it was smacked in postage, I think.
Right.
Welled up, and there was something wrong with it anyway. And it was completely swollen when I got it, and they said, "Oh, it'll be fine." And I kept it for about 4 months and used it, and it was absolutely fine. So,
Right.
I the same thing recently, and basically I could actually even feel the nutrient pack inside the swollen pack, so I know it actually hadn't burst. It just, and there's actually a Y yeast page which explains what's happened. That I think basically they've accidentally got some in there, so the yeast has gone off kind of thing and started to swell the pack, but it meant I couldn't actually smack it because it was so the outer pack was so taut, but it worked fine. So
Did you have to make a starter with those?
I do with everything anyway, just
Yeah.
because it's you don't know how healthy the yeast really is, how much I've got there, and I know now, like if I make a 1 litre starter for sort of regular ales up to 1060 or so, that's perfect for it.
Mm.
did you?
So-
Sorry, James,
Okay.
did you have a look what's in the thing? Because I've always had a sneaky feeling it's just a bag of oxygen and it just kicks everything off. So, did you hoik it out? And
Yeah, it's like a sort of browny liquid. It looks like a bit of work basically, and possibly some extra nutrients in it, but that's about it.
The other thing I had once was, what are the new White Labs ones, whether it's a pack within a tube within a tube.
The
And
pure
I had a
pitch
lot
stuff.
of yeast between the two tubes.
Ew.
And White Lab, no, it's White Lab said that's probably fine, but they did replace it in the end. But yeah, the inner one had leaked a bit. So, but yeah, I think that was probably just after they bought them out, and I haven't had any since.
So James, you were talking about having a house yeast. So if you did ferment with a beer, a yeast, and you found it suited your environment, and maybe it adapted to your environment, you could then save some of that yeast. And if you wasn't gonna, you don't brew every week. It's fine if you're brewing every week, you can just pitch from one brew to the next. But if you brew every month or 2 months or something, you could actually, via that process, take some of your wort and then pick a few cells out via that process to keep your own house yeast. And then you know, as it adapts, you could then save it again by the same process and have a sterile yeast to use that's adapted. So that's what that's good for.
I think one of the dangers from propagating from the bottom of a bottle and from slurry is that, okay, you might get a generation or two out of it, but you don't really know what is in that yeast. So in the yeast, you can get bacteria, but more importantly, you can get wild yeast. And as the generations go on, so those will multiply and will spoil the beer. One thing you can do, and I don't know if anyone has done it, is to acid wash their yeast slurry. That'll kill the bacteria, but it won't kill the wild yeast. So you'll get a perpetuation of wild yeast in the slurry into future brews. But acid washing is something that you could have a look at as well.
I'll let somebody else do the talk on that.
Did you ever, Phil, did you ever get a sense that when you recouped yeast from bottles, that it was more prone to contamination than with the slant process? Did you ever get a sense of a hit rate or?
Yeah, well, I only ever tried it 2 or 3 times, and one of them was a total failure.
Yeah.
And what I did used to do was, if I used a smack pack, I would pour a very small amount into, again, the sterile water, and then plate it up. So, you know, or take a sample straight from the pack and put it on a slope.
Yeah.
So I had a selection of a few different yeasts, so I didn't have to go out and buy another smack pack. And then I would grow it up via that process that was in the presentation. So you start with a very small amount and grow it up over 4 days to a pitching rate. And you know you hope you're catching it as it multiplies. So you tip it from one container to another, and it's actually multiplying at that point, not actually starting to ferment. You want to catch it while it's still fermenting and give it lots of oxygen by shaking it or actually using oxygen. But I didn't in those days, but you know, when I do get back to brewing, I will use oxygen again. I think that's really, really good for yeast health.
Because I did.
How long can you? Oh, sorry. I was going to say, how long can you keep a dregs in a bottle, and what's the best way other than in the fridge? I just finished one of these, and I've put star sand foil over the top. Well, how long can I keep that before it's not viable anymore?
I don't know.
Is it like a day? Do I have to do it, or will that last a month in the fridge? Or
well, from a bottle, I wouldn't give it a month, because you've opened it, you poured it out, and it's not going to be in the best of health, is it? Really, I would say a day or so, but I'm sure somebody will turn around and go, "Well, I've kept yeast in the fridge for 2 months, and it was used particularly, you know, produce very good beer. So it's depends what you want to take a risk for.
Yeah, get rid of the any beer and just store it under boiled and cooled water. It will last much longer. I mean, you're not supposed to, but I've stored yeast in the fridge for like 4 months or something like that, and it's still been viable. A bit slow to get started again reculturing it, but it's fine. If you store it under beer, it will go rotten in about 2 months. It'll start smelling rather disgusting.
Paul, that's actually not my experience at all. I think the longest I've kept the yeast under fermented wort or beer is for actually over a year in the fridge at roughly consistent 4C, and all you do is just whip up a starter, maybe step it up a second time and brew with it like no problem. And actually I have about 10 of them at the moment in the fridge, and like I only ever buy yeast if I'm using it for something specific. Like I've just been reculturing yeast over and over and over again for quite a few years now, and I've never had a problem with any off flavors so that I could detect, at least any like problematic fermentations, mold growth or anything of the sort. And
I normally grab some at the end of fermentation. As I'm kegging, I'll pour a bit into a sterilized glass jar, so it'll have tube and other crap in there. So it does that stuff does go stinky. If it was
I wonder if there is actually a difference here, because you use the yeast that has fermented a beer, whereas I overgrow my starters and I use the yeast that hasn't had the time to tire out, and that's the yeast that I use to grow again.
That's quite different, isn't it? It's a very
I think the crux is a completely different approach to collecting and keeping the yeast. Not in the way that it's stored, but what yeast is actually collected.
Yeah, I've done that. I've brewed with the yeast, kept a small amount of it back in something sterile, and then made a very small starter and used that for bottling. So to bottle and condition with the fresher yeast. So that's one way you can do that.
And also, don't know if I feel like we've all been talking about the standard Saccharomyces, but Brett is brilliant for that. Like I have actually majority of the yeast that I store is Brett, and that thing just keeps for years. Like nothing happens to it. Like I've opened a vial that I've been storing for 2 years, and it's now fermenting a beer right next to me. Like the lag time was just almost non-existent. It was a bit ridiculous. I was surprised.
But it's a different beast, isn't it?
Yeah, but that thing you can just store if it's sealed, and if it has at least something to munch on, you're looking at decades.
Yeah. Well, Brett itself, it munches on very little, doesn't it, in your beer when it's fermenting for 2 or 3 years. So
the other difficulty with taking dregs from the bottom of a commercial bottle is you don't know if you're getting the yeast they brewed the beer with or a dedicated bottling yeast. If the brewer's kind, they might tell you that very often they use a dedicated bottled yeast, which is specifically grown so that it will pack down tightly at the bottom of the bottle and will not cloud up when it's shaken.
Jeff, that's right, and there is a much better. You have much better luck getting the actual yeast the beer was fermented with, if it's like a mixed fermentation beer or lambic or something Belgian, etc. Somewhere online there is actually a list of multiple bottles. I think it's the Mad Fermentationist blog. So the guy keeps page of all the bottles where the yeast is actually viable and is the one from brewers rather than bottling yeast specifically. But I'm not sure if there is a source like that for Saccharomyces. I guess my interest is mostly lies in the mixed fermentation for growing dregs rather than more usual beers, if you will.
Jeff Cooper has a story about he collected a yeast from a bottle of wheat beer, and he made a very good lager because it was the bottling yeast which compacted down rather than the original strain. So yes, Paul, you do have to be careful. But things like
When you
Orval and 1845, they are the original yeasts. So,
And
There's
Worthington White Shield.
there's
sorry,
And Worthington White Shield, I think.
it did. I don't know about now. I have no idea, but it was at one point.
I've seen a spreadsheet that tells you whether the yeast is bottling yeast or the original yeast. It's been years since I looked at it, but I think there was something online that will give you a good idea of what's in each bottle conditioned bottle.
Was it based on any stats, or was it based on the inherent knowledge of what has gone into the bottle?
I don't know. I think just experience, really, whether people had probably a bit of both, right? Like the comparison charts. But I'll have a look, see if I can pull it
I
up.
think I posted a link to one of the homebrew UK forum or something, where they had one of the threads was exactly that. What was in the bottle? Was it the original yeast or a bottling one? Phil, when you do your first streak from your, I guess, your mother yeast source, wherever that comes from, when, let's say you did a streak from a bottle, presumably what crops up on the plate then would give you a good idea of how pure the yeast strain was in the bottle, wouldn't it? If there were clumps of weird stuff in there as well, or is that not?
That doesn't really show up. You need certain media to grow the yeast or whatever you've got there in to find out whether it's a bacteria or a yeast. It doesn't really show up that much. That's what I've been told. I've never come across it.
Yeah.
So whenever I chose any cells or clump of cells, it was always the neatest clump or clumps and the largest I could get without it touching something else.
Yeah.
So,
Okay.
you know, you did need different medium to grow up bacteria. So you streak it out and the yeast won't grow, but the bacteria will. So
Okay.
I don't know much about that. I just know that's how they do it.
Phil, if I can ask, when you mentioned previously about using some of the propagated yeast at the bottling stage, did you find that there was a significant difference with the quality of the beer when you added it at bottling for conditioning using the fresh yeast?
What, the yeast that I'd stored myself or using a fresh yeast?
Yeah, exactly. And then at the bottling
yeah. Definitely for barley wine. You can have a barley wine in condition in 2 or 3 weeks. You know, if you fermented a 9 to 11% beer, you know, we've got this competition coming up. So you put it in bottles. You don't want to put a tied yeast in there. You know, you're better off buying a brand new pack of yeast or saving it from your original starter to bottle it with. So it's a yeast that hasn't gone through that high gravity fermentation because it's going to be knackered.
Sure. Okay.
Basically.
You've also got like the specific bottling yeasts are quite often will only consume the short chain sugars, so they're there to basically carbonate the bottle and only basically take up the sugar that you've added for the purpose of carbonation. Whereas
so there's like specific strains that are just for that purpose, really.
Yeah, so that they don't continue fermentation in the bottle and either overcarbonate the beer or whatever, really. That's kind of the thinking behind them.
Okay, brilliant. Thank you.
Yeah, I think one example is the Saf Ale F2. I've used it a couple of times and it works really well.
Okay, great. Thank you.
I found a list, but it's a list of all the non-sac stuff that you can get from a bottle. I'm hoping James can post it. So Brett and bacteria and stuff that's unpasteurized. But I'm sure I've seen something similar with sac in days gone by.
It's in the chat there.
Thanks, James. Can't do it on my iPhone and my iPad. Too old.
Even with your glasses on.
Yeah.
Okay, is there any other questions or should we leave it there?
Well, I had a question. You were talking about high gravity beers, tired yeast. If you were introducing yeast to revigorate that yeast and carbonate, what kind of quantities would you need of yeast? So I'll tell you the backstory. I've had exactly that same problem.
I go back to what Paul asked me. When do I know there's enough, or is it enough yeast? Just guess.
Okay, so I'm in exactly the same situation, and I'm basically going to have to. I think I'm going to have to either wait longer, which I don't think is the correct answer, or B, uncap everything, and I'm thinking kind of rehydrate some yeast with a syringe. Is it kind of drops or into each bottle, or is it actually a significant?
Well, I know some people, they just use a few, they crumble some yeast in there. Never tried it myself. I've only ever done it with wet yeast, and what I did with one batch particularly, I had 30, 40 bottles and they weren't coming into condition. I bought a new smack pack and syringed out, worked out how much. I think it was like 5 or 2.5 mil. So I syringed out 2.5 mil and syringed in 2.5 mil of the starter, from the smack pack.
Yeah.
Put it in a dish.
Yeah, I think you don't really need much.
No, as long as it's active.
So when I was calculating once, I think I had to use like a gram or gram and a half for 20-liter batch. So basically, if you can just sprinkle 1, 2, or 3 those small
grains, yeah.
So it's really tiny amounts, like drop.
Yeah, if you use that F2 yeast, it's like you need like 2 grains of the yeast if you can pick them up with tweezers.
Okay. Good to know. Thank you.
Some people use the champagne yeast, the Jervin. Quite good for that.
Problem with that is you might, if you're adding it to a beer that's not had it in it, you might ferment more, end up fermenting further.
Yeah,
I've done that with a barley wine, and it does change. It changes the character. I use champagne yeast for bottling. But it changed the character of the beer.
At BrewCon last year, there was a guy from Fermentus that did a talk about stuff to do with yeast, and he said about champagne yeast that you shouldn't use it particularly for drying out beers, because it only ferments the shorter chain sugars, and it also secretes a killer enzyme that stops other yeast working.
Okay.
So you shouldn't co-pitch it, and using it to dry out beers is a bad idea compared to some other yeasts that you could find. So I don't know how. It sounds like it would probably be fine for a bottling yeast under that idea, but yeah.
Well, at the county beer makers, I mean, Phil, keep me honest on this. One of the recipes was to use a beer yeast with champagne yeast. I think the champagne yeast went in a bit after the beer yeast.
Most people put it in at the same time, I think, just for.
But that's what they would do, because back then there was only ye olde Nottingham yeast or whatever available. There wasn't a lot else. But now you use something like a 1728 and it'll just eat its way through.
I have a feeling that Curious Brew down in Ashford use a champagne yeast, but how they use it and where they use it, I'm not sure.
Yes, that's not far from me now. I must go and give it a visit.
The brand spanking new brewery, Phil. It looks quite good.
It's about 20 minutes away on the train.
Yeah, there you go.
After lockdown. Okay, Daz, any other questions, or should we leave it there? We can always come back if people want a thought of another question.
Phil?
Yeah?
Yeah, I got here late, so could you repeat all that, please?
No, you didn't. I saw you sitting down at the start of the talk. Don't. He comes in here, starts creating, making problems for everybody.
Very
Got
smart.
his suit on. Why have you got a suit on?
That's because he's got a tie on. He thinks he's in charge.
Collecting his prize for his last winning beer.
And he's had a haircut.
Yeah.
Yeah.
Oh boy.
Yeah, thank you, Phil.
Okay.
Phil.
Yes. Maybe another 10 years I'll do it again.
Phil,
There is a tradition brewing.
Phil, I'll make it 5 if I were you.
Okay. Yeah. Okay. No, I've been coughing a lot recently, so.
That's just flu, you know.